within the public libraries or an academic library. And you also, with this card, have access to like 3 million other titles across the state of Alaska because we belong to the Alaska Library Catalog. So there's a specific subject, uh, and we don't have a book on that or a particular book you need, or you're in a book club and you want to get a book, you can get it probably with this card. So I'd like to introduce Professor Morgan. She's our Instructor of General Studies for Arts and Sciences here at UAS. And you probably, I recognize some Morgan groupies in here because she is the person for natural science and local information on our flora and fauna. So welcome, Professor Morgan.
Thank you, Kathy. Thank you, Kathy, and thank you guys for coming tonight. I'm gonna actually. This is really bright with my eyes right there. Okay. Lana, does that still work?
Yeah.
Okay. I've been teaching here for quite a number of years. I grew up in this region actually. One of my goals in life is to learn everything that lives and grows in this whole area. Which I'm never gonna be able to manage all of that, but I'm gonna try. That means I get to be a lifelong learner. And I like knowing all the different things that live and grow in the area. I teach classes about that. About half of my work actually is to be teaching classes about such things. The other half of it here is to run the GED program and help people get their GED. So lots of stuff I have going on, which is a lot of fun. I have been kind of keeping track of what's going on with the water and the phytoplankton in this area for quite a few years now. I first started working for the university as the coordinator for the fisheries technology program. And part of what we wanted to do there was to provide support for the oyster oyster growing industry, which part of that help was to help them learn the different kinds of phytoplankton so that they could do some monitoring with a net, just do a quick little plankton tow, look at it underneath the microscope and see if they could see the ones that are toxic, Alexander and Catanella. And if they saw a lot of it, they might decide not to send their sample to the DEC lab in Anchorage to get it tested for paralytic shellfish poisoning, saxitoxins. It's an expensive process to do that, and if they could avoid sending a toxic sample up there and having everything like kind of shut down for a little bit, that was a benefit to them. So that's kind of how I got into learning more about the phytoplankton in this area, and it's kind of been a special interest of mine for quite a while. So,
Maybe while she's getting that, there are handouts that Professor Morgan
Yeah.
if you want.
over
Did
the
she
years,
send
I
that to
have
us?
taken. These are all pictures in the. Thank you, Lana.
The white paper.
I've taken pictures of various different kinds of diatoms and dinoflagellates, and then miscellaneous like ciliates and zooplankton and whatnot, and tried to get an example of every single species that would be likely to be in this area, and have quite a number of them. And so I put together an ID kind of sheet. There's several pages of it actually. There's a few I'm not actually finding quite yet, but maybe I will eventually. So there are 3 copies there on the back table by the candy bowl, and you are more than welcome to take them and use them. It's not something that you would need necessarily unless you have a phytoplankton net and a microscope to look at what's going on in the water here, but it's kind of fun to look at them because they're beautiful. And that is then just get turned on too. The good, the bad, and the beautiful. The good, I'm going to explain all these different parts of it. The beautiful is that a lot of these phytoplankton, the algae, microalgae, diatoms, dinoflagellates are really ornate and beautiful, so they're kind of fun. So what are phytoplankton? They are all single-celled. They're in kingdom Protista. Some of them can be single cells that are hooked together to make chains or colonies. That's still not a multicellular organism though, because there's no real communication between the cells. So it's just like a bunch of cells that are kind of hanging out together, not actually really being multicellular. Also known as microalgae, you need a microscope to see them. They live in the upper sunlit part of the ocean. Well, there's also ones that live in freshwater too, but I'm focusing on the ones in the marine environment here. So they live in the upper sunlit part of the ocean. And they are predominantly photosynthetic, although Some of them are not photosynthetic or strictly photosynthetic. There are some that also eat other things. Dinoflagellates will eat diatoms, for example, a lot of the time, but predominantly photosynthetic. They need sunlight, nutrients in warm-ish kind of water. They can't like super icy, cold water. They don't do so well in, but they're not plants, but they are similar to plants in that they need these different things in order to flourish and do well. So if you think of how well plants grow in the summertime around here, because we have more daylight, more sunlight, the air temperatures are a little bit warmer. You can see why there are more diatoms, dinoflagellates, and coccolithophores that actually live and grow and do their thing in the summer months around here because we have more sunlight, etc. Okay. There are several different kinds. There's groups of them: diatoms, dinoflagellates, coccolithophores, and this is what some of them kind of look like. So diatoms are a whole group of phytoplankton, and they are responsible for about 20 to 50% of Earth's oxygen production each year. That's kind of gigantic. They contribute greatly to the primary productivity of the ocean. They're basically the bottom of the food web. Dinoflagellates, in fact, actually eat them, and then other things eat the dinoflagellates. So dinoflagellates are not really part of the primary productivity. Diatoms really are. They are very important. They are single-celled. They can be in colonies. They have a cell wall made of silica, which is literally glass, silicon dioxide, and there's two halves of that shell that all of their soft parts are kind of held inside of. And often what you can really actually see underneath the microscope is the glass shell or frustule of the diatom. Kind of refracts the light a little bit, which can be really, really beautiful. It's kind of fun to look at things. I love microscope work. There's about 200,000 species of diatoms in the world. So I'm never going to learn all of them. I'm going to try. They're really teeny, from 2 to 2,000 micrometers. So the ones the net that I use to do phytoplankton toes is a 20 micron net. So I'm getting most of them, but not the very, very teeniest ones. You use a net that's much smaller than 20 microns, it gets plugged super easy, and then it's not really very effective. So here are some of the examples of diatoms we have in the area. I took all these pictures except for this rectangular kind of flower-looking one, which my sister took, and that's an unknown. I've not been able to identify it. I sent it to an expert guy, and he was unable to identify it because it's hard to know everything, but these are all pretty common. I see the Cattosaurus, these two right here. These are two examples of Cattosaurus. There's single-celled Cattosaurus, but a lot of them are in chains. And the Cattosaurus, each cell has a long kind of spicule kind of off of each corner of the cell. And so if they're all lined up, then they look kind of fuzzy along the edges, right? These are really common in the water here. I see them every summer. Wow. Thalassionemia has kind of like this folded ruler kind of looking arrangement to it. Eucampia, like really long cylinders that are stuck together with the inside parts kind of scattered throughout. Didelum is always in pairs of two, like this, together. Cocinodiscus is always round, and then it has like a pattern of holes all poked in it so that there's like water exchange. And then all of the internal organs kind of scattered around like that. Dictyocha is kind of this sprocket star kind of a looking thing, and those are pretty common in the area here too. And Pleurosigma down there, which is all kind of really long and tapered, and then also kind of bent a little offset from each side. So those are all kind of fun to see. The one here on the left-hand side, and there's three of them that are kind of overlapping, the ends overlap there, that is Pseudonychia. It makes domoic acid. And at bloom levels, if there's enough of it, it can cause amnesiac shellfish poisoning in people, which is not something you really want to get. And I'm going to talk more about that in a little bit when we get to the bad portion of things. So, there's more diatoms than this around here, but these are some of the ones that are the prettiest that I wanted to share pictures of. Dinoflagellates, again, some more pictures to share. They have two flagella that they use to propel themselves in the water. And the flagellum wrap around right here. This one you can kind of see. If I can get that. So that's their pointer. Pointer right here. So there's a cingulus, which is like around the whole outside at the equatorial line, basically. If you could say that, which it holds one of the flagellum, and then the sulcus, which goes down along the side, which holds the other flagellum. And sometimes, if you're playing with a microscope, you can kind of see the flagellum a little bit, even moving in there, but it's hard to get a picture of because it's so fine. And so the di means two flagella. So these are in the name two flagellum. They are two flagella, right? Many of them are photosynthetic, but some of them also eat other things, or some of them actually do both. They are photosynthetic, but they'll take advantage of being able to eat a random diatom as it goes by. Some of them are bioluminescent, which is pretty fun. And there's only about 2,300 species worldwide of dinoflagellates. We have quite a number of kinds around here. Starting off over there on the left-hand side, those are all different species of Protoparadinium over there, including the one on the bottom that has the two kind of long legs and the pointy top. Those are all different species of Protoparadinium. In this picture right here, there are all three different species of Serratium. So this is Serratium furca, this is Serratium longipes, and this is Serratium tripos. And those are common. All of the Serratium don't. They're just food for other things. Eat them, and so it's kind of helps the food web along. Proboscentrum is this guy right here. It's kind of a leaf shape. This one happens to be kind of a little more oblong. Some of the species are more round, but all of them have like this little flagellum off of the one end. To me, this looks like a leaf with the leaf stem. Okay. When we have brilliantly colored water around here, and I'm going to talk a little bit about how that comes about, the yellow or golden component of that is often this proboscentrum right here, like at bloom level, lots and lots of it. Not sure why proboscentrum does so well around here. Maybe it's just the years I've been doing this, I've been seeing quite a lot of it, but proboscentrum is one of the ones that can be pretty common here. These two on the right-hand side are two of the not so good ones. On the bottom right hand, Dinophysis makes okadaic acid, which causes diuretic shellfish poisoning. Not super common around here, but you don't want to get it. And then the real culprits for toxicity in this area are the Alexandrian catenella. They produce saxitoxins, and not just like one or two or five. There's like a whole suite of different related saxitoxins that are all nerve toxins, and they cause paralytic shellfish poisoning, PSP. So if you get these guys right here at bloom levels, you should not be eating any of the shellfish in the area. So more about that in a minute. Our last main group of things that we can get around here, not all the time. This is the third one. We've got diatoms, dinoflagellates, and then coccolithophores. Okay, they are autotrophic, which means that they photosynthesize. They make their own food by photosynthesis. So they are an important contributor to primary productivity. They're the very bottom part of the very bottom of the food chain. They all have a calcium carbonate shell, CaCO3, right? They produce oxygen when they are photosynthesizing, as things do when they photosynthesize. They produce oxygen, and because they have these plates that they have overlapping kind of shield-like plates all around them, made out of calcium carbonate, that the carbon part of that CaC, as these live and then die, they sink to the bottom. They're a little bit heavy. They eventually sink to the bottom. They become part of the snow that you see if you see deep sea videos, there's always like the snow kind of coming down. Part of that is the copolipivores. And that snow accumulates at the bottom, and so that is storing carbon in the bottom of the ocean, where it's just going to hang out there, not cause trouble. These guys are super teeny. They're spherical cells, and at most they're 100 micrometers. They can be as little as 5 micrometers. So it's very difficult to find them. I can still forward it, seems like. Oh, I see. You're going to fix me up?
Yeah, you want to change that.
I wanted to point out that each one of these plates is kind of a separate, like, they built them and then they have them all over around of their whole cells. And this is an image. I don't have any images of copolithophoros because they are too teeny for me to be able to get. I only have a light microscope. I have like a regular, decent microscope. This was taken with a scanning electron microscope, which doesn't use light. It uses higher technology. But it can get very, very clear, precise pictures of super, super teeny things. And so it would be awesome to be able to have one, but I don't know. Can you go to the next slide, please? This is a few more of them all together, and you can kind of see that quite a few of the plates can kind of start coming off. They are white. The calcium carbonate shell, it means that they are white. Okay, so, and that's important because, next slide. I'll get there in a minute as far as when they are, why that's important. The phytoplankton blooms are, so a lot of the time when I go out and do a plankton tow, and the things that I find, there's like several different things in there, quite a few different things, like 5 or 6 or 12 different kinds of things growing in there. And I'm like, oh, hey, it's some tripos, and then, maybe an Alexandrian cell or two, and some dinophysis, and like all different kinds of things. And then it's like finding all kinds of really fun Easter eggs to be able to like get pictures of. It's like, wow, I own a red one, really fun things. But then other times it's like all one species. Mm-hmm. Or like predominantly one species and then just a few other random things. It's like, wow, okay. So, and this is a picture I got of a bloom of the serration tripos. It was almost all the serration tripos, and then not really quite sure what this is. It might have been a worm of some kind. It was wriggling around. So there are, and I have a few pictures of different zooplankton at the end of my slides here because there are always zooplankton in my samples as well, which is pretty fun because they move around a lot and they're pretty fun. But you can get blooms of things, which means that like most everything that you see in the toe is a species, and they can be fine. This was fine. These provide food for zooplankton, which provides food for small fish and et cetera of the food chain. They can become a problem if you have a bloom of one of the ones that's harmful. So those are called harmful algal blooms. Mm-hmm. Traditionally, for a long, long time, people call them red tides because in some parts of the world, the ones that are harmful are red, even like brilliantly red, which is a great indicator. Oh, this is dangerous. Whoops. Around here, that's not the case. Alexandrium catanella is the one that is our biggest problem around here, and it is not red. It can be kind of reddish, brown, yellowish, kind of not very apparent at all. It's not really red. And the one we have around here that's red can actually look like literally like tomato soup on top of the water. It's not toxic. Wow. And so people know that I want to know everything that lives and grows around here and know some of this stuff. And so they will send me pictures or texts or messages, whatever, and be like, oh my gosh, the water is like, is it safe for my dog to go in the water? This is not, what's going on? And they kind of freaking out a little bit because the water can be a disturbing red, orange kind of color around here. And it's understandable that they're worried, but here, that one is not toxic. So color, not always a good indicator of like what's going on. Having a microscope and being able to do a toe and then look at it with a microscope is a good thing. So they are driven by sunlight. They're driven by warmer water temperatures. They're driven by nutrients such as iron. They are driven by the correct level of salinity, neither too saline nor too unsaline, right? And by what's eating them. So if something is in the area that is eating, like if you have a lot of goats, you can't grow good grass, right? The grass is never going to get really tall because the goats keep eating it. So you will not have bloom levels of some pretty common species, maybe, if there are a lot of other like zooplankton around eating them and keeping them from really reaching bloom levels. Sunlight, warmer water temperatures, and nutrients such as iron all can happen in the summertime. The nutrients such as iron come from the land, the terrestrial environment. So if you have a time period where there's like a lot of sun, a really warm sunny period, and then you have a rainy stretch, which is washing a lot of nutrients out to the ocean, and then it gets warm and sunny again, you can have massive blooms of things because the rain washes more nutrients out. Everything that was getting set up to grow well has just been fertilized, and then the sun's back out, water's a little warmer again, in the greenhouse is full of tomatoes, so to speak. Okay. Okay, next slide, please.
Okay, it's not working.
Did you give it back to me?
It's not working.
Okay.
If you want it.
So this is some of the water that people send me pictures or messages, and they're like, "Oh my gosh, have you seen the water?" And honestly, pictures aren't doing it justice because it doesn't photograph quite as well. And then we have a little more light here, but if any of you've been around for very long, you will remember that over the last few years, a few times, not quite every summer, but pretty close to it here for a little while, we've had water that looks tropical.
Mm-hmm.
And it's disturbing a little bit to some people because they don't understand what's going on there. And it might be dangerous. And people are aware that there might be harmful algal blooms, and they're concerned. So I get questions when this is happening. And so next one, you can also get intensely green water. Not quite as tropical looking, but like really intensely green colored. And the next slide. This is where we're going to talk about color mixing and why having the copolithophores be white is kind of an important component of all of this. Okay, so think of the tropical water, and so the components in that are going to be blue and yellow and white. And they're going to mix together to make kind of a turquoise, really vivid color, right? So the blue is reflected from the sky. We're kind of used to thinking of blue water. The yellow comes from these dinoflagellates. I took a bunch of, actually just made copies of the same picture to put up here because this is the prorocentrum I said was pretty common when these were happening. And they're pretty yellow, okay? And if you mix blue and yellow, you end up with kind of a greenish color, right? And you can see here, it took me a little bit yesterday, but found it online where you can take different shades of kind of blue down to like green. And at the top, you can see like viridian or forest green even. You can have kind of a bluish, greenish kind of a color, but it's so intense and saturated with that pigment that you don't actually see it very well, right? Not much light is getting into it to be able to like reflect back to your eyes. But if you mix white with it, then it really shows the color because the white will bounce back all of the light, really lighting it up and making it really apparent. And that's what's happening in the water in the summertime when we have that really vivid turquoise water. We have the blue from the sky, we have the yellow from the phytoplankton, of whatever kind. And then the white from the copolithophores just intensifies the color that's there by reflecting a lot of water back at your eye from the sunshine, really making all the colors pop. So, right, it's art. Okay, so the next color that we get around here is this orange, reddish kind of color. And this is the one that's like most disturbing to people because they hear it's red tide. Oh my gosh, this is scary. Women. Oh no. And so, then the next one, please. This is one of the more intense ones I've ever seen. And there was just a lot of it into the little cove where I was at. And there was a great blue heron and then a Canada goose that both came in and were kind of checking it out. And like, really, they neither one of them stayed very long because it was kind of weird, right? It's like, what is going on here? It was not, they couldn't see through it to be able to eat anything. It was just too much for them and they left. It's not harmful though. You want to go to the next slide? This is a it's a dinoflagellate. It's more of a flagellate, one flagellum actually. It's called Noctilucus scintillans, and it is bioluminescent. So in my bottle there, it was so thick I was able to just scoop the bottle up, and then it separated. The Noctilucus scintillans has a really high fat content, so that it does float to the surface where it can take advantage of the light. Which means that in my bottle where I scooped it, it rose to the top and separated. And so that top half of the bottle is solid Noctilucus scintillans cells. Underneath a microscope, they look like this. They're round. They have a flagellum, which they use to move around with and maybe to eat as well. Eat with, you can get picks things out of the water and eats with it. This is the one that if in the summertime you go down, there's like bioluminescence. This is the one that's causing the bioluminescence around here. Let's see, swimming in the bioluminescence is probably the most fun, but motoring or rowing or whatever at night and being able to see in the water, that's this guy. The scintillans is right, not the Luga, night sparkle. So the red during the day means that you should go out at night to try to see the bioluminescence, or phosphorescence, people call it. Not harmful. Okay. So we do have harmful algal blooms. This is the bad. We've talked about the good some. This is the bad, right? Harmful algal blooms are algae that produce toxins that are harmful to people and other animals as well. Marine mammals, seabirds, fish, and dogs. There are different types. There's amnesiac shellfish poisoning, diuretic shellfish poisoning, and paralytic shellfish poisoning. And speaking of dogs here, people have asked me if that red one that I was just talking about, Noctilucus scintillans, is bad for dogs. Is it okay for me to let my dog swim in it? It won't. I don't. There's nothing in it that should harm a dog, but also it does have a high fat content. I would not if it's really thick, your dog's not going to smell good, and it's going to really stick to their fur. So I wouldn't let your dog swim in it just for that reason, but I don't think it should harm your dog. Okay, so back to these. Go ahead. Amnesiac shellfish poisoning is so it's caused by domoic acid, which is a biotoxin, and it is made by the diatom pseudonychia, which in the picture here, these really long pinnate diatoms that are overlapping. When they are really the most active and the most likely to producing toxins, they're not just overlapping. They will actually slide past each other, and so they'll kind of all collect up into like a big stack and then past each other to be all stretched out again, and then collect all back up into a big stack and then past each other into a really long stretched out line again. And they'll just keep, I actually have spent quite a bit of time on more than one occasion watching them do this in a microscope. It's kind of mesmerizing watching them just move past each other into these different configurations. And when they're doing that, they're much more active than normal and are apparently more able to produce toxins then as well. So not really quite sure how that all adds up for them, but it's pretty fun to watch. They are concentrated in bivalves, as all of these are. They are accumulated through bioaccumulation in bivalves such as clams, cockles, mussels, oysters. You don't want to eat them then because they're not getting much in any one day. But through the process of bioaccumulation, they are storing all the toxins in their flesh, just kind of suctioning it off, right? And so then if you eat, because people have asked me too, if there's a big bloom, is it safe to go and even just like drink a cup of water? Can I swim in that? Is it safe? But when you eat a clam, you're eating all of the toxins out of hundreds and hundreds of gallons of water. Mm-hmm. And so they've done studies where they have determined that oysters can filter 50 gallons of water in a day. Similar, I'd be willing to bet for other bivalves. So if they're filtering 50-ish gallons of water a day, over the course of 2-week bloom or something like that, they've filtered hundreds of gallons of water. You're getting all of those toxins if you eat that clam. They can be passed on. The toxins can be passed on to Dungeness crab as well. If it's really super thick, it can also kind of accumulate in the gills of the Dungeness crab. And if you get ASP, amnesiac shellfish poisoning, it can cause vomiting, nausea, diarrhea, and abdominal cramps, headache, dizziness, confusion, disorientation, loss of short-term memory, motor weakness, seizures, respiratory secretions, cardiac arrhythmias, coma, and even possible death. And the short-term memory loss can be permanent. So this is not real common in Alaska. And I actually kind of, have there been any examples in Alaska, or is it really further south? Some discussion about that, but I do see, there's a picture I took of the Pseudonychia, which is responsible for the biotoxin, so it seems plausible that it's possible for this to be in Alaska. So, next one. DSP is caused by opideic acid, which is a biotoxin from Dinophysis. There's a number of different species we have here, also concentrated in bivalves through bioaccumulation, and it causes nausea, vomiting, abdominal pain, and diarrhea. It's not deadly, but it's not fun. A person can actually lose enough water through these processes, though, to maybe not be so healthy. You might need to be in a hospital while they keep you alive while you deal with this, but it's not like instantly deadly. So that's kind of good. Okay, and the last one, PSP, is the one that we do have around here, and it is a problem. And it is a naturally occurring biotoxin, a whole suite of saxitoxins, like I was saying, and it is caused by Alexandrium catenella. It is concentrated in bivalve shellfish through bioaccumulation as well, and can be passed on to both moon snails and crab. There have been examples of people who have died from PSP after eating crabs, and don't know if there's been any documented cases of anybody dying after eating moon snails, because people don't generally eat moon snails anymore. They have some native people have in the past, is my understanding. They are edible, but since they can have PSP, I'm not going to be eating them. The reason why they can have them is that their preferred food are the butter clams. They will bore a hole near the hinge and inject digestive enzymes into the clam, so that they can then slurp it up and eat it, right? So if the clam is toxic, the moon snail becomes toxic. So don't eat moon snails. If you eat crab, the way that you should, don't cook them whole. If you clean your crab before you cook it, you will not have PSP problems. If you cook it whole, and especially if you want to eat the butter, which is also the hepatopancreas, that's the most toxic part. Okay? You cannot get around cleaning the gills can have the toxins in them from just getting these all gummed up in their gills. Cooking the crab whole will drive those toxins into the flesh from the gills and from the hepatopancreas. So clean it, rip off the back, break it in half, get rid of all the guts, then cook them, and you're fine. So PSP is a nerve toxin that causes numbness, paralysis, and even death in the extreme cases. There are yearly incidences of this in Alaska, and that is caused by this little gorgeous dinoflagellate. So be careful. If you harvest and eat wild harvested clams, mussels, oysters, you are taking a risk, especially if you do so in the summertime. Mm-hmm. There's been a mnemonic that people have used for a very long time that it's safe to eat clams anytime except with any month with R, it's safe to eat clams. So that would leave May, June, July, August as unsafe months. And those are the least safe. Those are the ones that we have more sun, a little warmer temperatures, and a lot more likelihood of a big Alexandrian bloom. But it's not safe any time of the year. My sister and brother-in-law own an oyster farm, and the only time that they actually tested positive for saxitoxins was in November. they have a cyst resting stage where they kind of go dormant for the winter time. And those are still toxic as well. So if those get stirred up into the water column somehow, and the oysters are eating them, then they can be toxic any time of the year. Clams that are like in the mud flat, whatever, their siphon tube is right at the same level as those encysted um, Alexandrium cells. And so if they're eating them, then they are toxic as well. So be careful. Anything that you want to test, if you want to go harvesting clams and then be able to eat them, you can send a sub-sample of that to the CTOR lab in Sitka. The Sitka tribe has a machine that they will take your sample and blend it up and do an extraction and run that through the machine to let you know if it is safe or not safe. And if you hold the rest of your clams in the fridge until you find out, then if they're safe to eat, you can eat them. That's the safest way to eat wild harvested clams, is to send the, it costs something, but it's cheaper than maybe dying. Okay, so PSP, part of the bad part. Zooplankton, these are some examples. I don't know what all of them are. This is some kind of a snail. That's a copepod of some kind, right there in the top, kind of middle top. I think that this is a cyprid stage of a barnacle cyprid, maybe. The second to the left over there, all of these little tentacle this is all kind of moving, and all of these tentacles were moving. It was this big long string. I have no idea what that is, and this is kind of one isolated, like this big end looked like it might be the mouth. The whole thing was kind of moving. It was fun to watch for a little while. I have no idea what it was. So if you get interested in looking at things underneath a microscope, you can find all kinds of things which are fun to be checking out. And last, these are all beautiful. Some of my favorite pictures here. They can get the different red colors or whatnot that are coming. They're eating a lot of some kind of phytoplankton that produces a little bit of red pigments. But this guy ate so many of them that he's like accumulated a lot of that red, and it's really pretty beautiful. So this is this round one with the pokey part sticking out. It's a Cattosaurus cell that is coiled around, and I was looking at it from the top end, so you can see all of the spicules kind of sticking out. So anyway, these are some of my favorite ones to look at. They're all pretty beautiful. So endlessly fascinating. So thank you. And do you have any questions for me?
I'm curious, did the native peoples here have any traditions about not eating shellfish?
Mm-hmm.
Did they not eat them ever, or just not
They
eat
ate
summer?
shellfish, and then they had timing things. So like it was say, so don't eat them after skunk cabbage came up. Don't eat them when you start hearing the grouse hooting. And then it was safe again, like when salmon started running. I think it was something like that. So they had timing things depending what else was happening in the environment here. Those timing things are becoming a little less reliable as the climate changes some and as things change because of that. But yeah, they have traditionally eaten them and still do. The one case of PSP that I know about, kind of the one that I heard about that I was like, I don't really need to eat clams that badly. It was somebody from Metlakatla or visiting Metlakatla, I'm not really quite sure, but in Metlakatla ate clams, got PSP, was medevaced over here on life support, kept alive in the hospital here, absolutely paralyzed. And at one point, and I don't even know who this is, I don't know the person's name or anything, but was laying there in the hospital bed on life support, IV, everything being kept alive. And the doctors, nurses, whatever, were talking about the chances of this person living or not, whether or not they should pull the plug or not. And then they decided to not yet. We'll just see what happens. And then the person made it and relayed later that she could hear the whole conversation, but could not move a muscle, could not say anything, could not indicate in any way that they were not dead, that they were still there. And they kept the person alive long enough for the liver and whatnot to detoxify all of the toxins, which is kind of amazing. I was like, no, I don't think that that's something I need to do then. Um, and like I said, people like for a long time. I mean, I grew up in this area. The first time I was around PSP was in Myers Check, and everybody got out on a low tide and was eating clams. I was a child, and word came down. Everybody was that these are hot, so and so has lips that are tingling, and so everybody quit eating them. Then other people like will take a bite or rub it on their lips or something like that to see if it starts making your mouth tingle, and if it does, then you shouldn't eat anymore. I'm like, better than just eating them whole, but still a little risky to be doing things like that. And so, but part of the normal culture in this area, I'm not speaking specifically to native culture, but like people who've lived here and lived out in the middle of nowhere, Myers Check and whatnot, you just kind of try to happens. So, but if it's going to put me in a hospital bed when they're maybe talking about pulling the life support and I can hear them, I don't want to do that. That's not up for me. I'm not up for that. No, thank you. Yeah.
I'm thinking as ocean temperatures rise, and things are coming up in the current, and we're seeing the green crab. But what else possibly could be coming our way? Like what's going on in the Pacific Northwest? What little creatures are down there that haven't sort of uh, breached our waters?
And we might have more species that are going to be starting to be able to live here. And the definition of being a plankton, if you're a plankton, if you could get pushed around by the water currents, you're at the mercy of the water currents. And the predominant prevailing currents here come up the coast from about like Oregon, Washington level, and get swept up here. So anything that's happening down below us gets pushed up here, which is how the green crab got up here. The larva got pushed up here, right? And so as waters are warming, there'll be not only like the species that are already here will start like having more big blooms, but we might start getting more species coming in. That's a possibility. So far in Alaska, what we're seeing is we're not really seeing that many more harmful algal blooms in Southeast Alaska. We hear a lot about, don't go down and dig clams at Rotary Beach or Saxon Seaport or down at wherever Refuge Cove, because the butter clams are toxic or the blue mussels are toxic. We're hearing about it more right now because they're testing for it more. Okay, with the advent of CTOR setting up a lab at the Sitka tribe, and then they have agreements with like KIC and other tribes throughout Southeast Alaska. They're testing the public beaches for PSP when they didn't used to, and so we hear about it. That doesn't mean we actually have more, but further north in Alaska, there is more. And so there are massive seabird die-offs, and scientists don't know yet whether or not those are caused purely by starvation. A lot of the birds that are being found are emaciated. There are fewer forage fish for them to be eating, it seems. Some testing, though, on the flesh of the dead birds is also showing elevated levels of saxitoxins in the flesh, and so that might be a factor. And honestly, I would expect it to be maybe more than one factor. Is it PSP toxins? Is it starvation? Is it one of the things that warmer water does as well is just increases the animal's metabolism, and then they literally cannot eat enough to be satisfying that elevated metabolism. And that is being seen off the west coast of Alaska quite a lot right now. There's marine mammal die-offs as well, kind of for the same sort of reasons, and there's more harmful algal blooms. If you look at Alaska, like the back of your hand, the part where it's narrowest between us and Russia is having a lot of PSP blooms in that area, and actually even wrapping around up above the North Slope area has Alexandrian Catanella blooms happening up there, and which has never happened before. The water is much too cold, but the water up there is getting warm, so much warmer than it ever has been before, that they're having PSP outbreaks up there for the first time in like ever, whatever. They also have king salmon that are spawning in streams and rivers up on the north slope, which is new. So yes, climate change, Kathy, is part of the whole equation too. And it's good that people are aware of harmful algal blooms and the effect that they can have, to just be kind of careful about things. And right, if you hear about it in the news, it's definitely happening. So, other questions?
The microscopy here is just ordinary light microscope.
These are, yep, all of these pictures I took with my cell phone, various different ones through the years as I have them die. But the lens on the back, and I can just take it through the lens of the microscope. It takes some steady hands and like really fussing with it for quite a while sometimes, through an ordinary light microscope. Yeah. And I can sometimes get it all lined up and then also adjust the focus a little bit, and I can get it as crisp as possible in my pictures.
But the zooplankton are much larger than the phytoplankton.
That's correct. And that's a very good point. These aren't all the same size either. I have a tendency to want to be able to see everything and make it all kind of basically the same-ish size. And that's not realistic. This is much magnified and then also not proportionally correct with each thing in here. So the zooplankton are generally much bigger than the phytoplankton. Diatoms, dinoflagellates, copolithopores, they eat those phytoplankton. So, and I want to, the harmful algal blooms are a reality, and it's something that we are seeing more of in parts of the state, not so much around here, and it is a concern. I also want to remind everybody that while there are the harmful algal blooms that are kind of hard to think about, don't want to go there, primarily all the phytoplankton are very positive. They are driving the productivity of the ocean. They are the bottom of the food web. And everything that lives in the ocean then depends on them to be able to survive. So that's the good. The good are they're food for everything, or food for things that everything else eats, right? The bad is that the harmful algal blooms are not so good. And then they're beautiful. So there you go.
So in the upper right-hand corner here,
Yeah.
is that a colony of single-cell organisms, or is that?
It is.
Okay.
Yeah. So you can see kind of the chloroplasts and whatnot, kind of being like golden and green kind of a color. That's the center of the cell. And then there's the oval parts that are open there, but then they're kind of joined on either side. So those places where they're joined are two cells touching each other at those points. Yeah. Good question. Yeah.
So the harmful algae that the different crustaceans eat are not harmful to those species?
Right. Isn't that strange?
Yeah.
And then another whole level of that is that not only is it not harmful to the clams or the mussels or the oysters, which are like whatever, this is food, they actually can do a little bit of selective feeding too. They've done studies on oysters more, but they have an in-current siphon where they're taking in all this different stuff, and then inside they have internal organs, which actually can kind of separate out things and will just get rid of things that they don't want to eat. And they have a line of slime that they use to actually eat the things that they do want to eat. So they do some selection, but it's not based on toxic or not toxic. It's like size. I don't know how tasty it is. I'm not really quite sure. And so it's not only not toxic to them, but like sea otters can eat them and do eat them and don't seem to be affected by it. And they're trying to do studies to figure out what's going on. Some people that are observing with binoculars and whatnot, countries, think that they're noticing that they are selecting parts of the clam to eat and parts to throw away, which might be part of the thing, but I'm not sure exactly what's going on there. They may just not be affected by the toxin. Haven't determined that yet, though. Yeah.
Since you're finding them way up there where they've never been seen before, it's warming up. There's probably going to be possibly more of them in the ocean. Could that increase the percentage oxygen level in the atmosphere?
They are producing oxygen. So yeah, if there's blooms of ones that are really producing a lot of oxygen.
In other words, ocean warming could actually be producing more oxygen than we had before.
I think that potential is there. Yes, that the warming ocean could produce more oxygen than we've had.
And maybe the higher level of oxygen has something to do with the occurrence of fires, or is that too big of a stretch?
I don't know that you can make that link. I'm not sure that more oxygen is going to make more fires. You do
Because
need
if you've
oxygen to
ever
make
worked-
fires, but there's plenty in the atmosphere already.
If you've ever worked in a place where they have pure oxygen, like in tanks or anything, if you get that directed at a flame, boy, that will accelerate things.
It does. Yeah.
And so I just wondered.
Bridget, you had a question a little bit ago. Is that?
Well, I just, we hear on the radio over and over again about how the clams on this beach and that beach are, stay away.
Yeah.
How do like the oysters in that Hump Island Oyster
Yeah.
Farm, how do they avoid being
That's an
contaminated?
excellent question. So how do they avoid, like, any of the oyster farmers? Hump Island Oysters is one of the closest ones to Ketchikan here. How do they deal with this situation? And the way that how they deal with it is that they have their product in the water that's growing, right? The oysters in the water that's growing, and then when they want to harvest some, they take all of what they want to harvest plus a few more out of the water, and they store them in like big refrigerators, and then take a sample of that then, and to be certified to be able to sell them, you have to send your product to the DEC lab in Anchorage. And so you have to put your sample in a box with ice, blah, 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 because it has to actually get to Anchorage within a certain period of time, which is like 3 days, which isn't so much of a big deal from Ketchikan, but if you're like on the other side of Prince of Wales Island, 3 days is not very long. And it has to be within a certain temperature range when it gets there. So it's a little dicey getting your sample up there. Anyway, and they do what's called a mouse bioassay, is that they take the sample and they blend it up and they do some extraction kind of stuff and then they inject it into a mouse. And if the mouse survives, then it's safe. And if it dies, then it's not safe. And then it's also like not only if it dies, but then how long it takes to die to be able to figure out. Kind of. And so if they get word back that their product is safe, then they can go ahead and ship all of it that they've pulled from the water, ship it off to wherever they're going to be selling it to. If word comes back that it's not safe, it's toxic, then they have to put all that product back in the water so that it can get rid of that toxin eventually. Okay, it's called depuration, and shellfish can get rid of the toxins by depurating it. Different shellfish will take longer or shorter. Blue mussels get hot really fast and then also lose that toxin really quickly. Butter clams don't take up the toxin as fast, and it could take more than a year for them to totally get rid of it too. The depuration process for butter clams is long. So the oysters, though, oyster farmer, if they're toxic, they're not a loss. They just have to put them back in the water and let them continue feeding and doing all their metabolic processes, and eventually they will be non-toxic again, and then they can try it another time. Part of why, when I was developing this whole ID sheet and learning how to teach farmers how to identify Alexandrium in particular, if they could avoid sending a sample to the DEC lab when they were toxic, that was a money saver for them. So if they knew how to do a plankton tow, and if they saw a lot of Alexandrium catenella in their sample, they'd be like, okay, not sending these off this week. And then next week, try again, basically, and monitor, because then they would only be sending samples to the labs when they were more likely to be safe.
So the oysters are as susceptible to being toxic as the clams that are like right down in the.
That's correct. Yeah, so the clams and the mussels and the oysters are all bivalve feeders, so they're filter feeders. Sorry, their filter feeders are bivalves. So they're filtering all of these little guys out of the water and eating them. So yeah, they're all susceptible to PSP.
Does that include the gooey duck fisheries as well?
That includes the gooey duck
Even
fisheries
how
as
deep
well.
they are?
Yeah, they have to all of it. So the process with the gooey duck fishery in SARFA, I think, helped regulate this. Southeast Alaska Regional Dive Fisheries Association. They go out and they take some from the areas that are going to be open, and they send the sample up to Anchorage. And if it comes back safe, then they'll open the fishery, and they do that weekly. Pretty sure it's weekly throughout the whole season.
And then what about sea cucumbers?
Sea cucumbers are detritivores. They don't eat anything out of the water. They just go along the bottom and eating all of the
Like the algae growing in the
yeah,
pond?
all of the detritus that's accumulated on the bottom of the ocean. So they do not get toxic. Yeah. Good questions. Any more? Yeah.
So, can oyster farmers tell by a sample if the Alexander toxin is there?
They
for
can
them?
tell if the Alexandrian cells are there, which is a pretty good indicator that they might be toxic. It's not 100%. They can't say, oh, it's at bloom levels, it's going to be toxic, or it's like little below bloom level, so it should be okay. There's no like super hard, like level kind of correlation, but it's a pretty good indicator. Yeah.
But they're not doing anything to try to like protect them from.
No,
They're not taking any special.
there's
Okay.
no.
Yeah.
I mean, they have to. The waters here are really good for growing oysters.
Mhm.
This is a whole nother hat I have on. I used to work for Oceans Alaska about 15 years ago or something, and my sister and brother-in-law do own an oyster farm. I used to grow microalgae to feed baby oysters at Oceans Alaska, and so the waters here are really, really good for growing oysters. It's they're warm enough for the algae to bloom, but not too warm. Too warm, then you start getting some bacteria like Vibrio that are a problem, and there are a lot of wild phytoplankton for them to feed on. So they want them to feed on all of that wild phytoplankton. And even if they were able to like test and say, oh, they're really toxic right now, there's no way of keeping the alexandrium out and letting them feed on other things. You have to just be like, okay, we have to wait for this to pass. It's still a food source for them. It's not hurting them. It's just making them unsellable for the time being.
Mhm.
There's no reason why the oyster farming business here wasn't a booming business and a really solid part of our economy in the region throughout Southeast Alaska. They do really well here, and the market is not saturated. All right. We are like almost straight up 7:00. Supposed to be done at 7. Thanks, guys.
Okay.
And thank you for the UAS Ketchikan Campus Library for hosting Ask UAS. So will there be another one soon?
November.